Done, sir.
Vipran sir, please continue.
Okay, thank you, sir. So, good evening, our All India President Dr. Arvind Deshmukh sir, and all the present participants here. I, Dr. Bipans Kumar Tiwari, extend a warm welcome to all of you to this roundtable discussion on the topic microbial amylase. So let me begin with a brief introduction to microbial amylase, which are that among the most widely used industrial enzymes that play a crucial role in sectors ranging from the food processing to the pharmaceuticals. So microbial amylase are powerful enzymes produced by the bacteria and fungi that break down a starch into the simpler sugars. So unlike the plant or animal derived amylase, this microbial amylase are highly efficient, cost effective, and easy to produce in the large quantities. So these enzymes play vital role in wide ranges of industry due to their stability and the means under varying conditions of pH and temperature. So also we know that there are the different types of the amylase, alpha amylase, beta amylase, and glucoamylase are the most significant. So alpha amylase are the widely used in starch processing, in the baking, brewing, and the textile industry for breaking down the complex starch molecule. So amylase are essential in converting the starch to glucose and particularly useful in the production of the high glucose syrup and bioethanol. So microbial amylases are extensively used in the food industry for processes like the brewing, baking, and the sweetener production. And research continue to enhance their stability and activity using the genetic engineering by the immobilization and even the nanotechnology in the present research work. So the novel strains from the extreme environments are also being explored for the industrially robust enzyme. So as students, researchers, or the faculty member, this is the field where there is a huge potential for the innovation. From isolating or discovering the new microbial strains to engineering the hyperactive enzymes, there's so much left to explore, and this is the reason for that today we have chosen this topic. So now I request all the means faculty members, researcher or students who are present here or who have joined this session to share their means research experience, to share the insights, or even if any knowledge on the mileage, if you can put the comments. Okay. So now I welcome you all to share your experience. So just you can unmute yourself and share your research experience. If you have any knowledge or information on the microbial amylase, you can share it with the students, with the research and the faculty members are there. And also I request all the means members who are present here to write your name, affiliation, and if possible designation in the chat box so that we can connect with each other. Any faculty members, researchers, scholar, students who wants to share the research in microbial amylase, just unmute yourself. Shubhajit, Pooja, Amara, and one name is PPD. Name is not showing here. And then Amrita Pradhan, A. Sonia, Abhinandan, Amara, Brinda, Harshita Pujari, Pooja, Shubhajit, Bipin Chanda. Anyone can unmute, okay? It's just discussion. Yes, Sonia.
Yes, sir.
Yeah.
A pleasant good evening to one and all. Myself
Good
Sonia.
evening.
Am I audible to everyone?
Yes, ma'am.
Yeah, so I'm from Telangana. So my work is going on. Actually, I completed my identification part, sir. I collected soil samples from different places of Telangana region in the different districts from the different croplands like maize, cotton, uh, rice, like sweet potato. These all the crops and the groundnut especially, some uh, beans, delicacies. So here I collected more than uh, more than 100 samples from all the regions and different croplands. And finally, I found some amylase producing fungi. Actually, working on fungi. So here I got some clear zones around some fungal stains by using iodine test. So I'm using the starch agar media to isolate the fungi which producing amylase enzyme. Whatever the potential, whatever the clear zones they produce, so I isolated from each and every plate from the crowd and I preserved them and I sent for identification. Recently, I got the reports from identification, like Aspergillus species, especially different species are there, like Malbantia cinnamomea, Emericella, and Aspergillus terriers and Aspergillus guadeloupatus. So I want to go through with the special ones of any two of these organisms, which potentially produce amylase enzyme. Which then I want to check the optimization and the characterization of your organism and the cultural conditions for amylase producing organisms like fungi. Then I want to apply it for the industrial. So that is the specified latter of my work. And this is my work is going on now. Thank you everyone. This much I want to say.
Thank you, Sonim.
Yes, thank you.
Thank you for sharing your research.
Yes, thank you.
Now, anyone just unmute yourself. And any student, if you have any query to Saniam madam, you can ask, because this is the platform where you can clear your doubts also from the expert.
Can I ask you a question?
Sure, sir.
Did you quantify the amylase which is produced by the fungus?
Not it, sir. Further work will be going on that only, sir. Just
And
identify
what levels, what unitage you are looking at for quantification?
clear zones, zone of clearance, sir.
Zone of clearance is only qualitative.
Not. I did not go for quantitative estimation, sir. Just
Then how will you commercialize it if you can't quantify?
No, further step. I want to go for quantification.
Pardon?
Further, just the next level. I want to go for quantification.
Okay, because what happens is it's
I
commercial
completed only identification only.
You quantify.
Yes, sir.
You're restricting your research only to identification.
No. I want to go for industrial application first. Just now the PPHD completed and going with the identification.
So the first objective should be how much quantity it will produce.
Yes. One of the objectives.
And economically.
Yes, sir.
That's all. Sorry to have been bothering you.
No, sir. It's okay.
Thank you, sir. So here Amrita Pradhan, Abhinandan, Brinda, Harshit Pujari. Not research absence. If you have any knowledge on or information on that microbial amylase, you can share it here.
I want to tell few things.
Sure, sir.
Microbial amylases, many organisms are producing it. And as sir has told, not only the qualitative estimation, quantitative estimation is also equally important. And now, two ways are there for the production of amylase: SSF, and that is a solid state fermentation, and second by liquid fermentation. And maximum research is going on presently on thermophilic amylases also, thermoresistant amylases. So there are plenty of cases nowadays. As we know that bacillus are playing a dominant role in the amylase production. But my students also worked on amylase production via actinobacteria, because actinobacteria are relatively less studied as compared to other microorganisms. So I will give the name of my student who has worked on amylase, Dr. Krishna Bartake. He is from Maharashtra. He completed PhD under me, and his topic was amylase production by SSF, solid state fermentation. So I will give the email ID. I will give his contact number. Just I will type. Again, I'm requesting everyone, please type your name and type your email ID. The reason is that if anyone want to communicate, they will ask the question, they will be in touch. That is the main idea behind the discussion. To increase the communication, there is a communication gap among the research workers in India. By today's discussion, if only 3 participants come together and increased their communication, I will consider it is a success. So please type your name and type your email ID. Contact number is welcome, but it is not compulsory. If you are interested, you give your contact number. If not interested, don't give the contact number. So in this manner, now the maximum research is focused on the amylases application, as Bipran sir has told. It has a wide application in the industry. So because of the application, many students at present are working on the amylase production. Thank you. Thank you.
Can I add something, Dr. Deshmukh, with your permission?
Yes, you introduce yourself first to our audience.
Okay, this is Bhogle, and I'm fortunate to have been associated with Dr. Deshmukh since 1974. And out of all my career, I have spent about very easily 25 years in amylase production industrially by solid state fermentation. I think with respect to today's topic, this is sufficient. It's okay, Dr. Deshmukh?
Very, very true. Yes.
So
Yes,
anybody
yes.
having any doubt with little industrial experience I have, maybe I'll be able to
Yes.
help you out. I mean,
Yes, yes, yes. Very correct. So again, I would like to tell all Dr. Jayant Bhogle. He is a doctor, PhD, and from industrial side, because from industry, very few persons are doctor. Jayant Bhogle is one of them, and having wide experience of the fermentation. So regarding the fermentation particularly, if anyone want to ask the question, or communication even in the future. I will request Dr. Jayant Bogle, please type your name and email ID in the chat
Sure,
box.
if I am able to, I'll do that. I'm bad
Yes.
at computers. Yeah.
Oh, yes, yes, yes. So,
Or
will
they can get it through Dr. Deshmukh.
yes, or you can give, if possible, you can give your contact number also,
Yeah,
whatsoever
yeah, sure,
possible.
sure. And end of
And
the end
if
of the
anyone
session,
is
I'll do that.
interested, I
Yeah.
will share. I will share his contact number,
Yeah,
email
sure.
ID.
Now,
Okay,
now let us listen to the people who are working instead of
okay, okay,
telling
okay.
anything about me. Yeah.
Yes, yes. Okay. So, other PhD students working on this line can share their experience. Please.
Dr. Sandeep Jadhav has joined.
Yes, yes. There are 18 participants.
Yes.
If any one of them is working on PhD or MSc dissertation whatsoever, they can share their experience. Uh,
Even today
Chand
we
sir,
yes, sir.
yes, Biprans proceed because Chand has written he is working in Uttarakhand on amylases. Biprans sir, please continue.
Yes, anyone just unmute yourself and share your research. If you have any question, query on the SSF, you may put in chat box also. I can read out it. Or you may ask any query if you have.
Yes, good evening, President Sir, Professor Arvind Sir, and Bipran Sir. This is Dr. Prasoon.
Good evening, sir.
Yes, sir.
Mhm.
Just I want to put this on the table, this kind of imagination that I had while working on amylase. Uh, so we used to do agar plate assay, and then after 24 hours of bacterial growth, we used to flood it with iodine
Mhm.
to see the uh, hollow zone or the zone where starch hydrolysis has happened. Now, what I was thinking is, what will happen if I will use a colored, suppose jaggery or something else that is also having glucose or uh, some other substrate. So, in that case, iodine will not help. So, how can we detect such thing on kind of screening activity, agar plate or something like that? So if we are using a substrate other than starch or a starch that is already having some color, how can we detect that? So I heard someone is already working on amylase, so I will put
Mhm.
this question forward. Yes.
Can I attempt to answer your question?
Sure, sir.
Basically, your objective is to work on amylases, right?
Yes, sir.
Why do you want to use lower sugars then?
No, what I meant to say is my starch source is colorful, uh, brownish color.
So if
So, uh,
see
so
basically
raw
now
starch I want to use. Yes.
basically from your thing as the teaching faculty. Are you a teaching?
Yes, sir.
Hello?
Yes,
Yes,
sir.
sir.
Are you a teaching faculty?
Yes, sir.
See,
Yes, sir. I am a teacher. Yes.
uh, primary selection, secondary selection, and then the strain identification or whatever it is for a commercial application. Okay.
Yes.
So primary selection has to be done on a starch agar or something, and iodine is used. So that's okay. You don't get a blue color. See, what happens is if you are using a colorful starch, it seems the starch is already degraded to dextrins. That's why you're getting a colorful starch. See, when starch is when starch is degraded, there are some limit dextrins which are formed which give you a brown color.
Okay.
So, starch iodine complex, anything other than blue color indicates you that it's degraded. But if you really want to see the degradation, you should quantify it. Say,
Okay.
for
So for
example,
quantification, we need to use a standard starch that is colorless.
yes, soluble starch you get either from whichever company.
Mhm.
May make a starch solution, then add some buffer, acetate buffer at pH 5.3. Then it depends on draw a standard graph or titrate it of whatever it is. I mean,
Okay. Okay.
yeah, unless you quantify, that there is no fun in going further.
Yes. So after this qualitative test, we used to go do this the test tube based test
Yeah,
and
and if
quantify it.
it is
Yes.
if it is amylases, alpha amylase or something, you need to estimate maltose.
Okay. So in that case, what would be the substrate, sir? Maltose starch
Starch.
again. Oh,
See,
okay.
alpha amylase acting on
Will
starch
only break down.
gives you maltose as its end product. So if
Okay.
you look into the unit of amylase enzyme, it is micromoles of maltose produced from soluble starch at 5.3 pH in 1 minute at 37 degrees centigrade. Is 1 unit of alpha amylase.
Okay.
I don't know if you have idea about this quantification because there are very, very many methods. Novo
No,
has a
sir.
separate
I have not
method.
done. Yes, I used to do that early, just screening purpose. I did not quantify for
Okay,
alpha
okay,
male.
okay.
Yeah, yes, sir. So this was
Okay,
new
okay,
to me.
okay.
Yes. Uh,
Uh-huh.
but in
See,
anyway,
Novo
sir.
has a
Uh, uh,
different method of analyzing amylase. Then IP has a different method of analyzing it. JP has a different method of analyzing it. But all the three talk of quantification. How many units of amylase or how many micromoles of maltose? Say, for example, you have 50,000 units per gram. It means that if you take 1 gram of amylase, it will produce 50,000 micromoles of maltose from soluble starch at experimental conditions. Is it clear to you?
yes, sir. Yes, sir. Just
Yeah.
one more question. Yes, sir.
Huh.
This information is very actually mind opening for me. I wanted to know about the
Hmm.
industrial amylase. So for example, if Novo is already having it,
Hmm.
now what is the industry demand? If you know that, what type of amylases we should look for?
See, there are lot of companies producing amylase in India. Okay, Novo, basically you get imported. Biocon is producing, Advanced Enzymes is producing, and there are at least 5, 6 companies in Gujarat I know producing amylases and selling it for different applications.
Okay, so is there any research scope available, sir, or yes, like some kind of industry demand on which?
Any research, if it cannot be applied into commercial use, is only to produce papers.
Yes,
So you
sir.
first decide
And
what exactly you want to do, and then
so
go
for
ahead.
yes, yes. For so I am not aware of the industry demand, sir. That's why I am asking this. So if there is suppose thermophilic amylases
They
are also
they
worked on. Yes.
in industry amylases is used as our doctor has said right in the beginning, high glucose syrup, corn industry, pharmaceutical industry. You get all these digestive pills, right?
Yes, sir.
Unienzyme, lupizyme, aristozyme, blah 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 blah. All this pharmaceutical preparation contains amylases.
Okay. No, so is
Then
it
I
the
think,
cost of production is also already down, or is there any scope where we need to do some kind of research for improvement?
see, commercially, it is the input and output ratio which matters. If you are putting X, you should get 5X. Otherwise, there's no fun in running a fermentation industry.
Hmm.
Because the people from China, you got an enzyme complex enzyme from China. It's available at a throwaway price. 1 lakh units you are getting at 500 rupees a kilo.
Oh.
And then if you start using NLR grade starch or something like that to produce, you don't stand anywhere.
Hmm. Then okay. So this means it is a kind of saturated area already.
I feel so, but if researchers feel that there's a lot of scope, we can still go ahead because there's always room at the top.
Yes, yes, maybe in the application orientation, means application part, maybe
Yeah, yeah, yeah,
application of ML. Yes,
exactly.
sir.
So you talk to somebody in advance, they'll tell you what exactly it is. Talk to somebody in Biocon, they'll be able to tell you.
Yes, sir. Yes, sir. Thank you
Yeah.
so much, sir. Ah,
அப்ப படிச்சு எழுதட்டுமா?
So there is one question, sir, that how amylase can, from the Dr. Sandeep Jadhav, how amylase can be helpful in our daily life. I am not working on industry important enzymes, so just asking for enhancing the knowledge.
Enhancing the?
Knowledge, sir. Means he's not working on that particular amylase enzyme.
I mean, it's a very old technology. In Japan, it has been SSF has been practiced since 1950, 1960. In India, the first plant was started by Sankyo Japan way back in 1969. Then there were entrants like Biocon, Advanced Enzymes, and now very many people are there. It's basically a little saturated field unless you go for a very specific application. And people are talking of 1 lakh units per gram or so. That is the level of concentration they are able to achieve.
sir, I think amylase is also used in detergents.
Yes, yes, sir.
Yes. See,
Yes, sir.
I think some of amylases are used in textiles also.
Yeah.
Yes, sir. So I think Sandeep sir is asking for daily life. Maybe he's willing to know about it.
Digestive enzymes you get in the market, right?
Yes,
Aristozyme, lupizyme, unienzyme. Have you heard of these tablets, digestive tablets?
Yeah, union enzyme we have.
yes, sir.
So unienzyme was the first thing to be introduced in our country, and it was a collaboration of Sankyo and Unichem. The company was started in 1969. Earlier, Unichem used to import amylase from Japan. Then, looking at the demand, So that's how the first plant to make amylase started in India. Prior to that, this glucose companies, maize companies used to make that was bacterial amylase for their liquid glucose and other things. But for pharmaceutical, this is the first company we started. Then, of course, Novo was there right from day one. But it's a very, very, very competitive market.
Sir, one more question from your recent explanation that so which organism is now best industrial produce means is industrially being used? Is it bacteria or fungus?
See, if you want it for corn syrup, textiles and all, it is basically bacterial because there you get a thermo tolerant enzyme.
Okay,
Pharmaceutical, it is fungal based, which is normally Aspergillus oryzae.
okay,
Yeah, and what happens is solids deep culture fermentations as far as the yields and other things are concerned.
so for fungus, SSF is
And now the
better.
downstream has also.
For fungal enzymes,
SSF is
SS
better.
better. Okay, 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 sir.
Yes, for fungal enzymes. For fungal
Okay.
enzymes.
Mhm. What we usually
So
know that
whatever
SSF.
plants.
Yes, sir. Sorry, sir.
Pardon?
No, sir. You continue, please.
No, I
So actually, sir, I wanted
discussing
to
that.
ask.
It's not.
Sorry, actually, I interrupted. No, so in my mind, it was like SSF is hard to scale up. So this fact is new to me. That
What?
okay,
SSF is hard to scale up, huh?
scale up.
Who
Yes,
told
yes.
you?
So, sir, because
Who
tray
told
system,
you?
tray based system, somewhat
So
this.
if,
Yes, trays
pardon?
people use na for SSF. People use tray,
Yeah, so
no
if,
stacking
yeah,
racks. Yes, yes.
yeah, yeah, that's right. Yeah.
Okay, so
So
scaling
what's
up.
the problem in stacking the racks?
Okay. No, like liquid
We,
cultures,
we,
we can
we,
take
we have
10,000 liters or something, no, and then continuous fermentation can be done. While SSF, we need to do
continuous
tray to tray,
fermentation,
so
huh?
batch to batch. Yes, yes, yes. So batch to batch, tray will go, no?
My dear friend, deep culture fermentations are difficult to maintain. Sterilization, asepsis, plus you
Okay,
need to use a complex raw material like starchy raw material or which is little difficult to sterilize. Solid state fermentation is you have a variety of agro-based raw materials which are easy to handle.
okay.
I
So
have I
operating
have
cost is lower.
practiced it. Yeah, I have practiced it at 2 ton per day level.
2 ton per day. Great.
Yeah.
Hmm.
Oh,
How about sir?
Dr.
Yes.
Saab, Dr. Saab, Namaskar, Halabi Saab.
Good evening, sir.
Huh?
Yes, sir. Dr. Prakash Alami sir has joined, sir.
Yeah, it is nice to see him. Is he back from the US?
Not yet, sir. You give me some time. I will share my opinion after some time.
Okay, okay.
Okay, sir.
Hello, sir. Nice to see you.
Thank you.
Any participants, if you have any query, because today we are fortunate today with us, one expert is present here. Dr. Gokhale sir. So any PhD students or the student of UG or PG level, if you have any query for the mileage or even in the fermentation technology, you may put your query in.
All your research queries would be addressed by Dr. Halami. I am not the person to answer research queries.
I think that application part will be answered by you, sir.
Yeah, or scaling up. Research part, Dr. Halami is the king.
Dr.
And we are all fortunate to, yeah, we are fortunate to see Dr. Halami, despite of his busy schedule in the US, he has joined the group.
Yes, sir. He joined every
Hats
session.
off, Dr. Hats off to you.
Sir, thank you very much for nice words. And thanks to Dr. Tiwari also for remembering me. Just started small work here. Actually, microbiology and fermentation technology department in CFTI was a pioneer in several of the food enzymes, including amylase, glucosidase, catalase, of course,
Yeah, yeah.
proteins. Production, especially by solid state fermentation. And few years back, even I worked on some part of work on the amylase from bacillus, which was actually useful, especially for degrading starch and using as a that's degraded starch for as a carbon source. All right. You know that bacillus also has a vast diversity of amylase enzyme. And amylase has been extensively studied in a Bacillus subtilis with reference to its secretory mechanisms. The signal peptide of the alpha amylase has been engineered and studied the secretome of Bacillus. Tiwari Saab, can you hear me?
Yes, sir. You are doing.
Yeah, wonderful. Yes. And then this is what actually when I was there for the PhD work in Germany, we had actually a collaboration with Wageningen University in Netherlands, where they had actually the different mutants of the Bacillus alpha amylase. And what they found out actually is that how the signal peptide is useful for secreting from the intracellular into the medium or in the culture filtrate. Yeah. And then they had actually the mutant associated with the signal peptide specifically. And then we were looking with reference to the lipobox, the lipid modifying system, which is actually found in some of this kind of this peptide, which actually remains adhered to the cell wall because of the lipid moiety. And such lipopeptide which I have studied is from the bacillus is associated with immunity to the bacteriocin. Now, how this work actually the alpha amylase secretory system and then lipopeptide actually matters because the mechanism is one and same with reference to the secretion into the extracellular space. And you know that the most of the enzymes produced by the fermentations, and if they are secretory, if they secretes in the medium, has the additional advantage because through the fermentation we can actually collect them in the medium, concentrate rapidly, purify also. But when it is in the intracellularly made, then both the steps are actually becomes complicated for the user. Just to add few more information related to the alpha amylase, the diversity is concerned. Their catalytic is concerned, the substrate specificity. And this kind of actually was diversity. One can see actually the amylases from aspergillus. Now, 60s and 70s, several group from microbiology department in CFTRI has extensively worked on the alpha amylases from the fungal source and production by the solid state fermentation. Okay. And then this was actually the initial work which was carried out in CFTRI. If we say in the Bicon, the most of these enzymes were initially commercialized through the Bicon, the work that was carried out in CFTRI. Okay. Since I work mostly on the bacterial systems, we see that alpha amylase is very predominant or very commonly produced in bacillus, weakly produced in lactobacillus, right? If it is in a lactobacillus, the alpha amylase actually is useful. Why? Because it can be used as a probiotics again. Why? Because then this lactobacillus can be used in the poultry feed, and that poultry feed where this lactobacillus will be incorporated, will have the ability to degrade the starch, and that starch will be available for other microorganisms to grow. Okay. So if somebody says alpha amylase, there is no scope. No, it is not true. Hundred thousands of publications one can see on the alpha amylases, right? There with
नहीं नहीं,
reference
बट
to
हाला में साहब जब तक इसका क्वांटिफिकेशन नहीं करेंगे और इंडस्ट्रियल एप्लीकेशन नहीं ढूंढेंगे एट ए प्रॉफिट,
yes,
because
sir.
you know that there are giants in India also, like Advanced, Biocon, Novo has come in a big way.
Exactly.
From
So no,
Karnataka only, there are at least 4 or 5 companies making amylases for
exactly.
different applications. Poultry
Yes,
different, textile different,
yes.
pharmaceutical different.
Poultry
So,
feed, and feed.
so the challenge in front of research team is how to get a room at the top.
Yeah, exactly. So that is
So
what
if
actually we.
somebody is giving you 1 lakh unit, whether you can make 1.5 lakh units per gram.
Yes, yes. The substrate specificity, the specific.
Yeah, that's the specific. I mean, in talking in terms of commercial use or commercial application of profit for for the company,
Thermo.
if I am able to offer 1 lakh unit at 1,000 rupees, and if I am able to offer 1.5 lakh unit at 1,000 rupees, you will be put to an
Yeah,
advantage.
exactly.
Using same substrate only, you need to play with the organism. That is where the real challenge in the research lies.
Exactly. So how novelty actually we can look from the enzyme perspective, the fermentation parameter perspective, the
Yeah,
stability is concerned because there are now several methods you want to stabilize the enzyme, at a different
exactly.
temperature
Yeah, you
using the.
can you can you can get thermostable enzymes
Yeah, exactly.
or enzymes working at a low temperature, sacrophilic instead of only
Yes.
mesophilic.
So large
I think
number of.
we are together putting it in the right perspective to research people.
Definitely, sir. This is a
Definitely.
important area which actually connect the biochemistry, the fermentation technology, the industry,
Exactly. Yeah.
biotechnology,
Hmm.
and the enzyme, the basic subject actually could go for a more the crystallization studies also because there will be
Obviously, I mean, because one more thing happens, Dr. Saab, as you go on purifying it,
Hmm.
उसका stability कम होते रहता है.
Yes, yes.
And if you look at the temperate zone we are into and the pharmaceutical application, it should be stable at the room temperature till the end of expiry period,
Exactly.
especially into liquid formulations.
Yes, sir.
If you remember, Dr. Saab, we used to have takadiastase from
Takara,
Parker
yes,
Davis.
yes.
So they used to give enzyme sachets separately so
Mm-hmm.
that enzyme would be stable. Yeah. And then you add it just before you start consuming it and then consume the entire thing in 1 week's time.
Oh, wonderful. No, wonderful. Very useful information. So we can ask some other participant also about the reviews.
Yeah,
We have
definitely, bro. We are only speaking
yeah,
for
yes.
the last time. We would like to listen the research opinion
Hmm.
on this
Hmm. Maybe Tiwari ji
because
can go.
all along we are talking only on application.
You
We're
know.
talking only on application. The research portion we would love to hear.
Yes.
बराबर है भला में सब.
बराबर है.
So, Prasun sir, do you have any query, Prasun sir?
Yes, sir. Sir, actually, I am so much interested to learn more and more, and this Prakash sir is also there. One more question I would like to put up is regarding the downstream processing of amylases. Suppose alpha
Hmm.
amylase, if I want to purify,
Yeah.
so what is the best way to go for it?
You do a microfiltration, take out the water part of it and then spray dry. The other alternative is solvent extraction, which is costly.
Okay.
So nowadays the practice which is there is concentrate the enzyme, remove excess water, concentrate the volume and spray dry so that your energy in spray drying is less.
Yes, yes.
Because
Yes.
solvent recovery becomes uneconomical nowadays, looking at the cost of ethanol and all, and the regulatory aspects around that.
Hmm.
Yeah, just I
So,
wanted
No,
to
no,
include one more
yes.
thing.
you should get a precipitable quantity at least. That is at least 600, 700 units per ml in your broth. Below that, it will take 7, 8 days to precipitate out.
Okay.
Yes, Somapal Saha.
Yeah, just one more point I wanted to just add is with reference to the enzyme. If we are going for the no, food application, we just ensure that it is from the food grade organism, so that we don't run in a trouble when we use the total biomass or no or the crude preparation or partially purified enzyme, right? So we just ensure that it is from the food grade organism. That actually
Grass,
takes.
grass can be used.
Yes, yes. Grass status.
Yeah.
Thank you so much, Alamis and Gokale sir, for sharing your insightful ideas and experiences.
Thank you.
Thank you so much.
Thank you.
So, dear students, if you have any query, you may ask now because we are fortunate today with us two experts are here. One from industry and one from the academic.
Good evening, sir. Am I
Good
audible?
evening, Harshita.
Yeah, so this is Harshita Pujari. I'm pursuing my master's from Ferguson College, Pune. I had a doubt that if two different samples gives the same absorbance reading in a DNSA assay, for example, if 0.75 at 540 nanometer, does that mean both have the same amylase activity? And what if one sample reach that value in 5 minutes and the other in the 10 minutes? Like, how does the time taken and the other conditions affect how can we understand the enzyme activity?
See, if you are doing it by spectrophotometry, your glassware should be absolutely clean.
Okay.
Are you treating your glassware with chromic acid?
So I'm not carrying out any experiment, I just had a question, so I'm just asking.
Then do the experiment and then come back.
Okay.
There are a lot of theoretical things which we can talk on this.
Okay, thank you.
Any more question or query, or if you want to clear any your doubt?
Yeah, some questions are there in chat box, I believe, Tiwari ji. Now, what the student was asking, the theoretical perspective, yes, we agree. But now we have to, this is actually the time where we do the experiment, prove experimentally, and then come for the discussion is using this MBSI forum. So we'll be actually more happy to interact and then try to find out how best through MBSI platform we can actually help even the new researchers. All right.
Yes, sir.
तिवारी जी वो देखना कुछ चैट बॉक्स में दिखाई दे रहा है क्वेश्चंस.
There is only one question from the Dr. Sandeep Jadhav.
Yeah.
That was how amylase can be helpful in our daily life.
जादव साहब आप क्या करते हो?
His left, sir. He left, sir, meeting.
Then let him talk to us. Yeah.
So if there any question, so anyone, if you want to share your idea or research, just unmute yourself. This one is the opportunity to clear your doubts because we have expert today here on this particular topic. Okay, so thank you so much for joining today's session and for making it both engaging and informative. So I am especially thankful to Dr. Halamis and Dr. Gokhale for sharing their experiences and also their insightful knowledge with us. And we sincerely look forward to your presence in the next session as well. So thank you everyone for joining today's session.
Thank you. Thank you very much.
Thank you.
Thank you.
Thank you so much,
Thank
sir.
you. It was nice seeing Dr. Halami after a long time.
तो चलिए अभी क्या करें, मिलते हैं बाद में.
हाँ मिलते हैं, आप आ जाओ, यूएस से मिलेंगे.
Yes sir, definitely.
Yeah.
Thank you
Thank
so
you.
much.
Right.
Thank you, sir. Very